by the Deputy Vice Chancellor, Administration Professor Dalani Mohamed, the Chancellor to Gombe State University, His Royal Highness Alhaji Dr. Umar, Area of Gombe and Chairman of the Gombe State Council of Areas and Chiefs, being represented here Alhaji Muhammad Baba, Adali Gombe, the Deputy Vice Chancellor, Academics of the State University, Professor Rashid Abdulgani, the Registrar of the State University, Dr. Abubakar Bafito, the University Librarian, Dr. Henry Maigari, the Bossa of the State University, Alhaji Hassan Shehu, the Provost of the College of Medical Sciences, Professor Joshua Abubakar, Chairman of the Gombe State University Ceremonies Committee, Professor Maigari, our inaugural lecturer for today, Professor Fatima Maigari, the CED, the Federal Medical Center, deans, directors, HODs here present, the chairperson of the governing board of the Federal Students of Gombe, Haja Zainab Abubakar Ibrahim, distinguished professors here present, Professor Will Chekhov from Maltech, Yola, the past registrar of the State University, Mohamed Abdullahi Yehuda, all staff of the State University. We want to also recognize the presence of Honorable Aisha Maigari, the former Commissioner of Ministry of Education. We also want to recognize the presence of Mr. Umar Gombe. We also want to honor the presence, the husband of the inaugural lecturer here present, Alhaji Adamu Lamwa, Waziri National Engineer Dr. Said Maigari, the Latin from the Alhaji Farouk Maigari, Haji Zainab Maigari, Dr. Mariam Maigari. I want to also recognize the presence of the elder statesman here in our midst, Alhaji Yaya Amari, the former Vice Chancellor of the State University, Professor M.I. Umar, Vice Chancellor of Eastern University here present, other heads of institutions here present with all. We also want to respectfully recognize Dr. Umar, classmates, the lecturer, consultants, student Zoom students, members of the press, distinguished ladies and gentlemen, you're welcome to the University, and you're welcome to Bangladesh Hall, venue of the 25th inaugural lectures, the series of lectures of Gombe State University. Please, the Vice Chancellor, the invited guests at this moment, quickly I will invite for the introduction of the event the Chairman of the Organizing Committee, who is also the Chairman of the University Ceremonies Committee, Professor Mohamed Abdullahi, who will come to introduce the event. You're welcome.
The Vice Chancellor of Gombe State University, represented by the Deputy Vice Chancellor Administration, Professor University, the representative of the Chancellor of the University, and Adalam Gombe, the members of the University Senate, the inaugural lecturer this afternoon, Professor Fatima Maigari, and her family members here present, special guests, distinguished ladies and gentlemen. Let me, as they say, stand on the established protocol. It is yet another very interesting, special historic event for Gombe State University. Ladies and gentlemen, this university has been recording these professorial inaugural lectures at a very fast rate. When you go to some universities, you will find that they have been doing it for many years, and you find that they are still somewhere in the low numbers. Ours was within a short time, we are climbing and climbing very, very quickly. And certainly, this is the 25th edition of the inaugural lecture. The 7th from the Faculty of Science of this university, and the 1st from the Department of Biochemistry of the university. So certainly, as I say, we always make history. History is always made. And today, we are yet here again to listen to Professor Fatima Umar Maigari, a professor of clinical biochemistry and nutrition, who is going to talk on small molecules at the biochemical architecture of human health. And I believe I have had the privilege of perusing through the lecture, so I would like to assure you, whether you are in the sciences or the medical college or in the arts and humanities, you will find this lecture very, very interesting, very educating, and certainly no one, I believe, will go out here without taking away something useful, whether in terms of issues of nutrition or human health. So I believe she is going to profess on this, and certainly we are going to make a lot out of that. Mr. Vice Chancellor, let me not maybe digress a lot into talking about what Professor Maigari will talk about. But certainly, as I say always, inauguration lectures are traditions universally in universities, where professors come to give some kind of account of what they have been working on, and probably they make postulations as to what trends are existence and where the future lies, especially in their own fields of specialization. So I believe this afternoon we are going to hear about this and many other things from the inaugural lecturer. At this juncture, let me please invite the Vice Chancellor to come up for his opening remarks before we go properly into the business that has made us gather here this afternoon. Mr. Vice Chancellor, sir.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم. Your Excellency, the wife of the Executive Governor of Gombe State, here present, the Deputy Vice Chancellor Academics, Professor Rashid Abdul Ghani, the Registrar of Gombe State University, Dr. Abubakar Aliuba Peto, the University Bossa, Alhaji Hassan Shehu, the University Librarian, Dr. Abare Maina, former Vice Chancellor of this great university, Professor Umar, visiting Vice Chancellors, especially from the Northeastern University, Professor Yusuf, a very good friend of Gombe State University, representative of the Chancellor of this university, then Adalan Gombe, the Chairman Gombe, the Chairman of Ceremonies Committee of Gombe State University, Professor Muhammad Guramaduku, special guest of honor, Hajia Zainab Abubakar Ibrahim, Chairman, Management Board of the Federal Teaching Hospital, Gombe, members of State Executive Council here present, distinguished Royal Highnesses, temporal and spiritual, deans, directors, head of departments, our special inaugural lecturer of the day, Professor Fatima Umar Maigari, Maigari's family here present, especially the former Commissioner for Education, Professor Aishatu Maigari, Dean of School of Postgraduate Studies, Directors, Head of Departments, members of the press, distinguished gentlemen and ladies, good evening. I stand here to represent the Vice Chancellor of this university, Professor Sani Ahmed Yauta, who is unavoidably absent due to some official commitments. I want to welcome you all to this great event. It has been a tradition in Gombe State University that every professor is expected to come and have a special day on his or her experience as a professor. We want to appreciate our founding fathers who have actually made this inaugural lecture series to have started, and also for the present administration for sustaining it. So today, I want to invite everyone to listen closely to what Professor Aishatu Maigari has for us. Small molecules as the biochemical architecture of human health. I'm sure by the time we leave this place, we are going to learn a lot. So as I wish the inaugural lecturer all the very best, I also want to wish us a very happy stay during the lectures. Thank you very much.
Thank you, Prof. Give a round of applause once more for The Vice Chancellor Administration, Professor Umar, representing the Vice Chancellor. Please, our inaugural lecturer for today is Professor Fatima Umar. Thank you very much. Please, at this moment, distinguished ladies and gentlemen, while I invite Professor Fatima Maigari to the stage, I will have the singular honor of inviting Professor Isaac. Professor Isaac is the University auditor, and he also serves as the Dean of Student Affairs of the State University. Please, Prof, you will come for the reading and the citation of the inaugural lecture.
Your Excellency Ma, the wife of the visitor of Gombe State University, the Vice Chancellor of GSU, ably represented by the Deputy Vice Chancellor Administration, Professor Danladi Umar, the Deputy Vice Chancellor Academic, Professor Abdul Rashid Abdul Ghani, the University Registrar, Dr. Abubakar Bafeto, the University Librarian, Dr. Henry Abari, the University Bossa, Mr. Hassan Shehu, the former Vice Chancellor of GSU, Professor M.I. Umar, Officer of the Order of the Niger, the Vice Chancellor of Northeastern University, Professor Yusuf Hamoda, revered traditional rulers, our respected Professor J.W.D. Somasundra. Vice Chancellor, permit me to stand on the existing protocol. I have the singular honor and privilege, with the authorization of the Vice Chancellor, to render the citation on Professor Fatima Umar Maigari on the auspicious occasion of her inauguration as a professor of Gombe State University. Born into the family of Yeriman Hashidu and Serkin Malamanduku through Alhaji Usman Maigari Gombe, Professor Fatima Maigari was educated at Air Force Nursery and Primary School, Victoria Island, Lagos, Aliyu Ibn Abi Talib Primary School, Kano, and thereafter at Hassan Ibrahim Gwarzo Secondary School, Kano, where she obtained the West African Senior School Certificate in 1999 and a diploma in data processing and IT at Inter-System Computer Institute, Kano. Professor Fatima Maigari had her higher education at the famous Bayero University, Kano, where she obtained the Bachelor of Science degree in Biochemistry in 2004, Master of Science degree in Biochemistry in 2009, and the terminal degree, the Doctor of Philosophy in Biochemistry, in the year 2015. She further earned a postgraduate diploma in education from the National Teachers Institute in 2011, a certificate in Arabic proficiency at Gombe State University in 2019, and a Master of Public Administration, also from Gombe State University in 2021, demonstrating a breadth of intellectual progress that transcends disciplinary boundaries. So Professor Fatima is among very few professors with 2 master's degrees. Professor Fatima Maigari joined the services of Gombe State University in May 2010 as an assistant lecturer in the College of Medical Sciences and was posted to the Department of Biological Sciences before the official takeoff of the college. Through sheer intellectual merit and a commitment to scholarly excellence, she rose through the ranks to Lecturer 2 in 2013, Lecturer 1 in 2015, Senior Lecturer in 2017, Reader in 2020, and was proclaimed a professor on 1st October 2023. From the beginning of her career, she held several departmental responsibilities as follows: Level Advisor, Registration Officer, examination officer, project coordinator, field trip coordinator, and SciWest coordinator. These roles helped build in her a rare combination of administrative competence and care for students. In addition to her primary responsibilities at Gombe State University, Professor Fatima Maigari has served as a visiting professor at Saadu Zungur University, Bauchi in 2024, and as an adjunct professor on pro bono appointment in the Department of Chemical Sciences, Northeastern University, Gombe since 2023. She has served as a pioneer technical advisor for the establishment of MBBS program at the Lincoln University, Malaysia in Kumu, and the establishment of the Jibril Aminu College of Medical Sciences at the Federal University of Kashere, Gombe. Professor Fatima Maigari's contributions to institutional development at Gombe State University cannot be overstated. She was appointed acting head of Department of Biochemistry in 2017 and subsequently served as substantive head of department from 2020, overseeing both the Department of Biochemistry in the Faculty of Science and the Department of Medical Biochemistry in the College of Medical Sciences, simultaneously, a dual mandate that speaks volumes of the trust reposed on her by the university authorities. So she was a double head of department in GSU. She played an instrumental role in the establishment of degree programs in the College of Medical Sciences and the Faculty of Pharmaceutical Sciences, working assiduously through successive accreditation exercises for the MBBS program by both the National University Commission and the Medical and Dental Council of Nigeria. Her efforts culminated in the program securing full accreditation status and an increased admission quota, a milestone achievement with far-reaching implications for the training of healthcare professionals in the Northeast and Nigeria at large. Also, during her tenure as head of department, Professor Fatima demonstrated a bold vision for academic expansion. She pioneered the introduction of 5 new academic programs in the department, which are a postgraduate diploma in biochemistry, master's degree in biochemistry, and the PhD program in biochemistry. She was instrumental in the takeoff of the BSc in Human Nutrition and Dietetics and the BSc in Biotechnology. These additions have significantly broadened the academic offerings of GSU and created new pathways for students to pursue careers in the life sciences. As Deputy Dean of the Faculty of Basic and Allied Medical Sciences, Professor Fatima extended her institution-building efforts, further contributing to the establishment of degree programs in nursing sciences, public health, and pharmacology, disciplines with direct bearing on the health and well-being of the peoples of Gombe State and beyond. In January 2025, Professor Fatima Maigari was appointed Director of the Directorate of Research and Development of this university, a fitting appointment for a scholar who has long championed the culture of research excellence. Within 1.5 years in office, her impact resonated and was historic. She facilitated access to the institutional-based research fund for more than 90 researchers drawn from different faculties and departments across the university. This singular achievement has galvanized a new culture of research productivity and given dozens of academics the motivation to pursue impactful scientific and human-centered inquiry. I was also privileged to get the IBR under her leadership. She further introduced a monthly webinar series to foster intellectual exchange and research visibility across the university community and organized a capacity building training workshop for 100 university staff. These initiatives reflected her conviction that research development is not merely a function, but a culture that must be deliberately nurtured. Over a career spanning more than 15 years, Professor Fatima has taught an impressive array of undergraduate and postgraduate courses. Her classroom is known for its intellectual rigor and her approach student-centered pedagogy. She has supervised over 16 postgraduate students at the master's and doctoral levels, a responsibility she undertook with remarkable thoroughness. She serves as external examiner to Bayero University Kano, Yusuf Maitama Sude University Kano, Modibo Adama University Yola, and Abubakar Tafawa Balewa University Bauchi, spreading her expertise widely across the northern region. Professor Maigari's research interests are broad and impactful, encompassing nutritional biochemistry, antioxidant studies in chronic diseases, ethnopharmacology, and medicinal plant biochemistry, environmental biochemistry and toxicology, and food science. Her work is deeply rooted in addressing the biochemical dimensions of health challenges that afflict the communities of Northeast Nigeria and beyond. She has published 35 peer-reviewed articles in reputable national and international journals, and contributed a chapter to a Springer Nature edited volume on sustainable agricultural waste management. Her research has attracted 2 IBR TED Fund grants. In Gombe State University, Professor Maigari Fatima is a member of the University Senate since 2017, member Senate Business Committee, member University ICT Committee, and member Vice Chancellor Search Team representing Senate in 2019. She was also the Secretary General of the GSU Muslim Umar Shura Committee between 2020 to 2022, a member of the GSU Strategic Planning Committee, among others. She is a UNICEF-certified psychosocial support master trainer and school-based management committee master trainer, having facilitated numerous trainings across Gombe State. She is a member of the Gombe State Committee on Food and Nutrition, the Gombe State Education Enrollment Drive Campaign Committee, among others, for want of time. Professor Fatima Maigari has served as a National Universities Commission resource person on verification visit to multiple universities across Nigeria. Professor Fatima has received awards of recognition within Nigeria and beyond Nigeria. She received another award for her dedication to education in 2017 at the 4th International Conference on Medical Science Technology by the University Kuala Lumpur in Malaysia. Professor Fatima is a member of the Nigerian Society of Biochemistry and Molecular Biology and the Chemical Society of Nigeria. She has rendered invaluable service as a peer reviewer to several international journals, including the Asian Food Science Journal and Journal of Scientific Research and Report, and the Asian Journal of Food Research and Nutrition. Professor Fatima is happily married to Ambassador Adamu Ibrahim and has a daughter, Aisha Nawal. Let me, on behalf of the Vice Chancellor, invite this cerebral, prolific, and distinguished professor to profess to this important gathering that is hungry to hear her. Thank you for listening.
The Chancellor of the State University, ably represented by Ada Gombe, the Deputy Vice Chancellor, the Deputy Vice Chancellor Administration, the Deputy Vice Chancellor Academics, Visiting Vice Chancellor from Northeastern University, Gombe, the Registrar and the Principal Officers of the University, the Chairman, Governing Council, Federal Teaching Hospital, Gombe, the CMD Federal Teaching Hospital, Chief of Gombe and the Attorney General of Gombe State, the Provost College of Medical Sciences, heads of departments of medical biochemistry and biochemistry, Dean School of Postgraduate Studies, the Director Institutes of Education, other heads of departments, distinguished members of Senate, members of the university community, my uncles, parents, family and friends, and my father watching online, wife of the governor and my head girl, Waziri Hashidu, the Dalatun Gombe Abba, my dear students, members of the press, invited guests, ladies and gentlemen, good afternoon. It is with a sense of humility and gratitude to Allah subhanahu wa ta'ala for giving me the strength and opportunity to stand before you today to deliver this lecture. I wish to express my sincere appreciation to this great institution for providing the platform upon which my academic career has been nurtured and developed. This is the 25th inaugural lecture in the series of professorial lectures in the history of our university, and it's a privilege I bear with full awareness of its weight. You may wish to note that the progress of modern medicine, nutrition and biotechnology rests on the intricate understanding of molecules that are invisible to the naked eye, from glucose to cholesterol, from amino acids to vitamins and minerals. These small molecular entities govern the structure, function and destiny of the human body. This lecture today explores a paradox that while molecules are small, their consequences to human health, disease, and survival are immense. This lecture is both a scientific exposition and a personal reflection on more than a decade of teaching and research in the field of biochemistry at Gombe State University. And it is my sincere hope that this lecture will inspire students, colleagues, policy makers, and the wider community to appreciate the field of biochemistry in addressing the present life and health challenges. So before I delve into my lecture today, I'm sure most of us know the common saying, a hungry man is an angry man. Why? So for a start, when a person is hungry, there's a hormone, a neurotransmitter that is called serotonin, which is low in the body. And this serotonin hormone helps when it's low, it increases the anxiety of a person. A person will be anxious. Insomnia, a person can't sleep. Most of us, we can't sleep when we are hungry. So it's because this serotonin hormone is low. It causes depression and headache. So sometimes when we are hungry, we tend to have headache. How about when we eat and our stomachs are full? This serotonin level increases. It's been secreted in the body and it's called the happy hormone. So all this anger will be no longer there. So it promotes the feeling of happiness and influences learning and memory. Most of my students here will believe when we are hungry, no matter what we are being taught, we don't understand that. So what is the discipline of biochemistry? Biochemistry was first coined in 1903 by a German chemist. It deals with the chemistry of life at the cellular and molecular level. And this life may be the human body, animals, plants, or microorganisms. At its core, biochemistry explains how atoms form molecules and how these molecules interact to form cells, how these cells develop to form tissues and organs which together comprise the human body. In summary, biochemistry is the only language the human body understands, just like computer understands the programming languages and algorithms. So biochemistry has multiple dimensions. In pathology, biochemical clues help the physician understand and diagnose diseases. A patient may present himself with stiffness or pain in the small joints, and on biochemical evaluation, it may be confirmed that there's elevated uric acid level, confirming gout. In biochemical pharmacology, the knowledge helps understand the mode of action, the toxic effects of drugs that we take, some vasodilators, some local anesthetics, and antibiotics. We are all familiar with the drug omeprazole, which is used in the treatment of peptic ulcer. So this omeprazole helps in inhibiting a membrane-bound enzyme hydrogen ion ATPase in the gastric mucosa, so lowering the hydrochloric acid within the gut. In agricultural and plant sciences, biochemistry deals with photosynthesis, nitrogen fixation, and the knowledge is needed to enhance the yield production, the prevention of plant diseases, and in testing pesticides and other contaminants in the soils, the food, and other products. In nutritional sciences, biochemistry is essential for understanding the effect of various cooking methods on food nutrients, and also in designing the nutritional composition of diets, especially for the vulnerable groups such as the children, pregnant, lactating women, and the athletes. Industrial biochemistry is another field which involves the application of biochemistry for the commercial production of various biomedical substances such as our hormones. The common one is the insulin hormone, some vaccines, and growth hormones. This field of biochemistry is now well established and called biotechnology. In medical sciences, biochemistry can be applied to help one understand the biochemical changes resulting in all the physiological alterations of the body. Biochemical architecture in this context refers to the organized and hierarchical arrangement of molecules within this living system. Just as a building depends on the beams, bricks, rods, and mortars, the human body depends precisely on the organization of these small molecules. So this architecture in this context operates at 3 levels: the structural levels involving the atoms, the molecules, metabolic levels involving the enzymes, and the regulatory levels involving the hormones and signaling molecules. So these small molecules in the body are the elementary unit of metabolism and cellular communication. They do not merely participate in life processes, but they define them. And what are they? The first one is carbohydrate. We are all familiar with carbohydrate, 6 classes of food. Number 1, carbohydrates. So carbohydrate is a small molecule and the most abundant biomolecule in nature. We are all taught since primary school carbohydrate is energy-giving food. So it serves as the primary source of energy for the human metabolism. Glucose, as you can see here, is one of the smallest carbohydrates in nature. It's a monosaccharide and it occupies a central position in most biochemical pathways. It's the principal fuel for the brain, erythrocytes, cornea, and for cellular respiration. We have the different sources of carbohydrates such as the maize, the rice, the sorghum, and so on. Another dietary carbohydrate is sucrose, what we call table sugar, the white sugar. So this sucrose is a disaccharide used as a sweetener for our tea, our coffees, our juices, and all other beverages. A teaspoon of this table sugar provides approximately 17 kilocalories of energy, and it's composed of glucose and fructose. So glucose and fructose comes together to form one molecule of sucrose. So after digestion, it's been hydrolyzed to form glucose and fructose. Lactose is another carbohydrate that is found in milk. So each milk, so the previous slide. So when we take our milk, it contains a carbohydrate. So milk also is not a source of protein, but also a source of carbohydrates. So this lactose is composed of glucose and galactose, as you can see it over here. So this lactose mostly appear in urine in the early stages of pregnancy because it's synthesized by the lactating mammary gland. Starch is another plant polysaccharide. We are all familiar with starches. Most of our food are starchy foods, and it's the main dietary source of carbohydrates. Another carbohydrate is cellulose. Most of these fibers that we consume, the spinach, the vegetable soup, the cabbages, are sources of cellulose. So cellulose is the most abundant plant structural polysaccharide. This one is found in the plant cell wall. That's why the texture of plant is different from that of humans. Mostly ruminants and sheep, such as our cows, our goats, they can eat these grasses and survive because they have the cellulose producing enzyme, and it can be digested by these animals or ruminants. However, human beings, we cannot digest it, so there's no dietary calorie for these fibers, for these vegetables. However, they serve as dietary sources of fiber, promoting bowel motility, slowing glucose absorption, and so on. Another class of amino acids, another class of molecules are the amino acids. These amino acids come together through what we call peptide bond to form a protein. Another class of food is protein apart from carbohydrates. So the biological functions of each protein depend on the chemical properties of the amino acids, the linear sequence of the amino acids, and the tertiary structure of these amino acids. Amino acids are basically classified as essential because the body cannot produce them, and we have to consume them from other sources of food, the meat, the beans, the milk that we consume. This particular structure is the human insulin. Most of us are familiar with the insulin hormone, especially the diabetic patients. So this is the structure of insulin hormone. As you can see, it has two chains, the A chain and the B chain. Each cycle, I don't know if it's visible, is one amino acid. If you look at it from down, we have threonine, lysine, proline. So depending on how they are arranged, depending on the activity of that particular insulin hormone. You can't change the positioning of this amino acid. Once you change the positioning of this amino acid, then you have ultimately altered the function of insulin, and that person can be diabetic. Another protein that we have is the elastin. This is a connective tissue protein that have a rubber-like quality, and it's flexible. When the skin is pinched, I don't know if you can pinch your neighbor or pinch yourself. So when the skin is pinched or bloated, elastin in the skin aids in the skin going back to its normal size. The concentration of elastin is more in the child and the youth compared to the person of old age. That's why when you pinch an old person, it takes time before the skin goes back to its normal position. The uterus also contains lots of elastin protein. During pregnancy, that's why it increases the stretchability of the uterine muscle, especially during childbirth. I think the gynecologist will tell us more. It's also the protein that is found in the lungs. When you inhale oxygen, the lungs expand. When you exhale the carbon dioxide, the lungs shrink. When you inhale again, the lung will expand. Why? Because of this elastin protein. Another protein, what we call the fibrous protein, is called alpha keratin. This alpha keratin is found on our hairs and the nail. For the ladies going to the salon to comb their hair, they are altering with the alpha keratin. That's why the hair seems to be longer, because it's changing all this dimensional structure of the protein. Another protein is the immunoglobulin and the albumin, called the globular proteins. Hemoglobin is the common one that we know, which is a globular protein of the red blood cell. And it tends to chelate iron. If you can see the iron within, Fe. So iron is being chelated within the hemoglobin. As you can see, it has 4 different types of chains. The alpha, 2 alpha chains and 2 beta chains. There's heme, and iron is attached within this hemoglobin. So hemoglobin also is a globular protein. So beyond their structural tools, amino acids also are precursors to different hormones, such as neurotransmitters, such as the serotonin I made mention, which is the happy hormone. We have the thyroxine from the thyroid gland, which is secreted or synthesized from tyrosine amino acid. Another small molecule that we have is lipid. We know lipid mostly as our fats and oil, the dietary fats and oil. So these lipids constitute the fundamental structural matrix of all biological membranes. So all the cells within the cell membrane contains these lipids, and they provide the concentrated form of metabolic energy. So when you eat more of the fat, the energy that is being produced in the body is more compared to that of carbohydrates. So these lipids also help in the absorption of what we call fat-soluble vitamins. About 90% of dietary lipids are made of the fats and oil. As you can see, it's called triglycerol. This is just the chemical structure over there. It's fatty molecules of fatty acid attached to a backbone of glycerol. Cholesterol is another form of lipid. Most of us are familiar with cholesterol, and it serves as a substrate for the synthesis of many steroid hormones, the bile acids. It's important for membrane fluidity and the list of steroid hormones as we have them here, the aldosterone, testosterone, estrogen, and so on. Among the lipids also we have what we call the transport lipids, which are the lipoprotein. They help in transporting different things within the blood system. So in addition to carbohydrates, proteins, and lipids, another set of small molecules are the vitamins. Yes, the polyunsaturated fatty acids. Most of us, I believe, we are familiar with the omega fatty acids. We are meant to believe the omega fatty acid is the good fatty acid. Yes, we have the omega-3 fatty acid, the omega-6 fatty acid, which is the niacinolic acid, and the omega-9 fatty acid. Why are they called omega fatty acid? I went somewhere and someone was advertising her product to me, telling me it contains omega fatty acid, and she's trying to explain what omega fatty acids are. So, as you can see, this is the structure of omega fatty acids. Omega is the last alphabet in the Greek alphabetical system. So counting the COOH is the first carbon, and CH3 is the last carbon. So when you start counting from the omega carbon, which is the last carbon, as you can see, this is CH3-1. The end edge here is another CH2. As you can see, the third one, the third carbon contains a double bond. That's why it's called omega-3 fatty acid. If you can see for the omega-6, if you count it here, 1, 2, 3, 4, 5, 6. So on the sixth carbon atom, there's a double bond, making it omega-6 fatty acids. And most of these fatty acids are found in our olive oil. That's why mostly we are encouraged to eat olive oil, the fish oil, the sesame oil, and they tend to be expensive because they contain these omega fatty acids. So the next small molecules I made mention are the vitamins. These vitamins and minerals are needed in small or minute quantities. However, their roles are very essential to the functioning of the human body. We have what we call fat-soluble vitamins, which are the vitamin A, vitamin D, vitamin E, and vitamin K. So this vitamin A participates in vision, and it's also good for the immune system. The vitamin D is good for calcium metabolism. Vitamin E serves as an antioxidant vitamin fighting different infections. And vitamin K, blood clotting. When a person gets a wound, vitamin K helps in healing that wound so that blood will stop, the flow of blood will stop. We have other water-soluble vitamins, which are the different, what we call the B complex. Most of us are familiar with the B complex, especially those taking vitamin or mineral supplements. These are the vitamins that are involved. So these B complex are very important, and they participate in over 400 enzymatic reactions within the body. The most common or famous vitamin is the ascorbic acid, vitamin C. I believe you all like vitamin C when we were small. So this is the structure of vitamin C. As you can see, the chemical structure over here, and it's very critical to collagen maturation, heme production, and enhancement of the immune system of the body. The other ones are the minerals, especially the trace elements. They are important to the human architecture. Calcium is important for bone formation, muscle function, and nerve signaling. Phosphorus is important for energy production. We have sodium and potassium for maintaining the blood fluidity activity in the body. Iron is essential for red blood cell. We've seen how iron is chelated within the hemoglobin. Iodine also is necessary for the production of thyroid hormones. We have what we call a free radical. This free radical is any species with at least one on and unpaired electron in its outermost shell. They are highly reactive, and once formed, they start a chain reaction. Their danger comes from the damage they do once they start this reaction, because they react with most cellular components such as the enzymes, the DNA, or even the cell membrane. So there are also some reactive species classified as reactive oxygen species. We have the hydrogen peroxide, the hydroxyl radical. So when the production of these oxygen species is much more than that of the antioxidant system in the body, oxidative stress develops. So these molecular injuries accumulate once there's oxidative stress. And the underlying pathogenesis, next slide, the underlying pathogenesis of many diseases, among it is aging. Oxidative stress causes aging, immune dysfunction, diabetes mellitus, cancer, allergy, and other numerous neurodegenerative disorders. However, the body produces some major antioxidant enzymes. We have the superoxide dismutase, the catalase, the glutathione peroxidase. Some of these vitamins, such as the vitamin C I mentioned, the vitamin E, and the vitamin A, which we mostly call beta-carotene, that is found within plants, all serve to protect or have this antioxidant property. So what happens when there is disturbance in this biochemical architecture? Let's start with carbohydrates. The human body maintains the blood glucose level within the narrow fasting range of 3.9 to 6.1 millimole per liter. Chronic elevation in this blood glucose, either arising from insulin deficiency, that is type 1 diabetes mellitus, which is common in the children because there's deficiency of this insulin hormone, or insulin resistance as it's in the case of type 2 diabetes or gestational diabetes. This particular elevation of blood glucose initiates a cascade of biochemical disturbances known as glucotoxicity. So this glucotoxicity produces the microvascular complications of diabetes, such as the neuropathy, nephropathy, diabetic retinopathy, atherosclerosis. That's why when a person is having chronic diabetes and gets wounded, it's very hard for the wound to get healed. As well as the macrovascular complications. Mostly diabetes comes together with hypertension. If you see a diabetic patient, mostly they have hypertension because of these macrovascular complications. Additionally, most of these diabetic patients and obese individuals are advised to lower or restrict the consumption of table sugar, the white sugar that we have. Next slide, please. The white sugar, and be advised to take alternative sweeteners such as the aspartame. Why? Because this aspartame contains two amino acids, aspartate and phenylalanine. For the table sugar, after it's digested, it produces glucose and fructose, ultimately increasing the blood level of glucose. However, for this aspartame, it's all just amino acids, so it has nothing to do with blood glucose level. Another alternative sweetener is the saccharin. This saccharin has lower calorie. For the table sugar, it's about 17 kilocalories per teaspoon. Count the teaspoons that you put in your tea. 17 kilocalories. But for the saccharin, it's just 0.2 calories per teaspoon of this sugar. And mostly in Hausa, we call this saccharin mayan sugar. And it's about 300 to 400 times sweeter than the normal table sugar. The next one that we have is the stevia. I believe most of us now are familiar with this stevia. This stevia is a natural plant-derived sweetener, and it has 0 calories. So no matter the amount or quantity of this stevia leaf, the sugar that you take, it has no impact on the blood glucose level, and it's sweeter, almost 300 times sweeter than the normal table sugar. As you can see, it contains carbon 20 sugar. Lactose that we have discussed, which is sugar in milk, remains indigestible in some patients having lactose intolerance. So what happens when this lactose cannot be digested? Because they don't have this enzyme beta-galactosidase. The lactose remains unabsorbed in the intestinal tract, as you can see it. So once it's unabsorbed, the bacteria within the intestinal tract tend to ferment the lactose, producing carbon dioxide gas and hydrogen gas. This tend to form these clinical symptoms of either diarrhea. Maybe you have had someone complaining that when they take milk, it gives them diarrhea, or flatulence, lots of flatulence or colic pain. So this is as a result of deficiency of the enzyme beta-galactosidase. In patients with galactosemia, the galactose in the lactose fails to be converted to glucose. What happens? This galactose tends to be accumulated within the blood and tissues of the cell. And once it's accumulated, it goes into the lens of the eyes together with its alcohol sugar, what we call galactitol. So accumulation of this galactose and galactitol within the lens of the eyes is what causes eye cataract. If you've ever seen someone with eye cataract. So when light comes, instead of it to be straight, it will be divergent within the eyes, and it causes clouded vision. And when it's chronic, it even causes blindness. So maybe some of us have exercised once or twice in our life history, or have certain vigorous physical exercise. The demand for energy, let's see the next slide. So when we exercise, we tend to need more energy, especially for our muscles. What happens, especially if you are running faster? After running, you tend to you breathe, needing more oxygen because the body gets more oxygen. The glucose that is stored within your body is converted to pyruvate, as you can see on the right-hand side here, to acetyl-CoA in the normal state before that exercise or vigorous physical activity. So this ATP, producing ATP is the energy currency in the cell. It's called adenosine triphosphate. Just like in Nigeria, we recognize Naira. In the United States, we recognize dollar. The body energy, all this energy we are saying is in the form of ATP. However, when this exercise is much, instead of it to produce acetyl-CoA, it goes in the other direction to produce lactate. So this lactate tends to be deposited in the muscles. So you start by running fast, you get tired, you slow down to walking fast, you slow down to just standing or even sitting. And at the end, you have muscle pains. Why? What causes these muscle pains? Because of the lactate that is being deposited within the muscles. So it causes the muscle pain, contributes to fatigue, and reduce muscular performance. So a rise in the concentration of this product also is what we call oxygen deficit or oxygen debt, and that's what you mostly see in asthmatic patients. Because of this oxygen debt, they have to use the inhaler to inhale because they need more of this oxygen. If not, lactate will accumulate and other biochemical and physiological conditions may develop. Regular exercise, however, can enhance the body's capacity to manage lactic acid levels, making it a valuable tool for athletes. We've seen mostly these athletes that do doping, they tend to inject themselves with steroid. So it increases the threshold for the hemoglobin to hold on to oxygen. So this particular biochemical pathways continues unlike the others. We all know deficiency of protein causes kwashiorkor. And obviously it's the deficiency of these essential amino acids because the body cannot produce them. Okay, sorry. Let's go. So this is dental caries or dental cavities. Most of our foods, the drinks, the sugar and the sweet that we consume, when we close our mouth, there's no oxygen. So the bacteria within the mouth, we call them Streptococcus mutans, they act in the absence of oxygen. So they produce this lactic acid. The lactic acid produced just like in exercise tend to affect our normal tooth, causing cavity. However, most toothpaste advertise their products as good. They prevent cavity because they have fluoride. Fluoride is a metal which tends to inhibit one of the enzymes in the previous pathway that we have seen. So lactate will not be formed because there's no glycolysis. A single amino acid substitution, next. A single amino acid substitution is what causes sickle cell. Maybe you've seen a patient with sickle cell anemia. So what happens is just a substitution of one amino acid, just as we have seen in that arrangement of amino acid in insulin hormone. Just one amino acid, glutamate, right from transcription, just as you are writing, you make mistake. Instead of writing Fatima FA, someone writes FO. So that single mistake, it was changes glutamate to valine. And as you can see, the normal hemoglobin has a disc shape, that particular one. So the disc shape is the normal hemoglobin with glutamate. Once it has valine, it tends to have this sickle-shaped hemoglobin, and it's rigid, and all the complications of this sickle cell patients develop because it cannot carry the normal oxygen as the previous one, and the lifespan of the red blood cell, instead of it to be around 120 days, which is 3 months, it takes only 10 to 20 days. Yes, next slide, please. Albinism. Maybe you've seen albino once or twice. What causes this albinism? It's just a deficiency of one enzyme, converting tyrosine amino acid, as you can see, into melanin. This tyrosine amino acid is converted to dopaquinone and subsequently to melanin. This melanin is responsible for the color of our skin, the hair, and the eyes. That's why when we are exposed to sunlight, because it tends to absorb the UV in the sunlight, we become darker. When we stay indoors for some days, we become lighter, because the more you get to the sunlight, the more the melanin is being formed. So in this particular patient, the tyrosinase enzyme is absent. So the pathway for the production of this melanin is abolished. Subsequently, they have no color, and it tends to affect even the eyes. That is why they have this photophobia, and they are affected by sunlight. And the skin is mostly exposed to cancer or other skin diseases. We have been meant to believe that smoking is dangerous to health. All these smoking pamphlets, they'll say they are dangerous to health. Why? We've seen the amino and the protein, elastin, which is on our skin and the lungs. So for those smokers, the smoke, once they inhale the smoke, it tends to activate the production of an enzyme called elastase. So this elastase tends to break all this elastin within the lungs, so it's no longer as stretchable as it is before, and it may lead to what we call chronic obstructive pulmonary disease. For all these allergic reactions, some of us will say we are allergic to this, we are allergic to that. Next slide, please. All this allergy is as a result of the production of histamine, mast production of histamine from the mast cells. So when a person is exposed to anything he doesn't like or any allergen, once the body recognizes it, it converts histidine, which is amino acid that we see, that small molecule, to histamine molecule. And this histamine is responsible for the teary eyes, the swelling and inflammation, runny nose, and all other symptoms that is being experienced in allergic conditions. Another metabolic substance is what we call the creatine, which is synthesized from 3 amino acids, and it's found mostly in the blood, the muscles, and the brain. It's used as a source of energy. So once it's needed to be excreted out of the body, it will be converted to creatinine. So this particular creatinine will be exposed or excreted in urine. However, when there's some exogenous or endogenous causes, depending on what happens to that particular individual, the secretion of creatinine is lowered and its level rises within the blood. And subsequently, when the test for creatinine is done in the blood test within the hospitals, it shows a significant amount of creatinine, which is an indication of kidney failure. Mostly we've seen jaundice, what we call shawara in Hausa. So this hemoglobin, that is the red blood cell, once it needs to be degraded, it will be converted to bilirubin, a very long and complex biochemical pathway. So this bilirubin will be eliminated in the urine. Once there's a problem, this bilirubin can no longer be effectively eliminated outside the body in the urine, and the bilirubin is yellow color. So once it gets deposited within the blood, it causes that yellowish of the eye, the lips, and all the symptoms in these people with jaundice. We have these newborn babies whereby jaundice is common because their liver is not yet matured to eliminate this particular bilirubin. However, it can resolve naturally, but when the excessive production or accumulation of this bilirubin occurs, it can affect the liver. That's why mostly in the hospitals you see when a baby is having jaundice, you'll be placed under blue light, what they call mercury light. So this bilirubin will be converted to another product that will be easily be excreted in the urine and will not affect the baby. We have what we call atherosclerosis or coronary heart disease, which is as a result of high level of cholesterol. Because when we eat more of the fat, especially when the fat is what we call the bad fat, that is the low density lipoproteins, it has high level of cholesterol, and this cholesterol tends to be deposited within the arteries. This is the blood arteries. You can see the red blood cells passing. The yellow is the cholesterol. Once they are being deposited, it reduces the flow of this blood from the heart to other tissues, and therefore the person might develop coronary heart disease. The concept of hidden hunger describes a population that consume adequate calories yet insufficient micronutrients. So we have the deficiency of vitamin A. I don't think that. Another slide, please, not this one. Deficiency of vitamin A causes. Yes. So deficiency of vitamin A mostly is associated with night blindness, poor immune system. We have deficiency of iodine causing goiter, this makoko. That's why our salt is being mandated to be fortified with iodine. We have deficiency of vitamin A, which decreases the utilization of calcium in the body, and for zinc it decreases wound healing and affects the DNA. So the other structure. All these molecular networks, the other one, sorry. So this is all molecular network. I just explained them individually for didactic clarity, but they exist within the cell as a network. So anything that is being affected affects another one. So all these pathways are within our cell, and each dot that you see is an intermediate that is being produced. Each dash represents an enzyme. As at October 2025 last year, the International Union of Biochemistry and Molecular Biology identified 600 enzyme catalyzed reactions within the cell. So for any disease, one of these pathways is being altered. So the cascading nature of biochemical disturbances explain why early detection and intervention is critical, because by the time clinical symptoms manifest, a molecular cascade has often been proceeding for years or decades. Biochemical biomarkers measured in blood, urine, tissue provide a window into these processes long before irreversible structural damage occurs. Nature is the oldest and most expensive repository of biological activities. Long before the advent of synthetic pharmaceuticals, humans across all continents relied on plants for the treatment of infections, inflammations, metabolic disorders, and pain. These traditional practices were rooted in empirical observations accumulated over many years. Plants synthesize this secondary metabolite just as defense mechanism against all those pathogens, herbivores, and so on. These same compounds, when used either ingested or applied medicinally, tend to interact with all these biochemical pathways that we have seen in ways that can be therapeutic. Example, we have the salicin from the willow bark, which gave rise to aspirin, quinine from the cinchona bark, and artemisinin from Artemisia annua transformed malarial therapy. So the lesson is clear here. Nature's molecular library contains solutions to human diseases that remains to be discovered and validated. However, this therapeutic exploration of medicinal plants must be balanced by rigorous attention to safety. The natural origin of a compound does not signify or guarantee its safety. So some of the researches that have been conducted, I think we've done some on glycemic index. So this glycemic index just measures how a carbohydrate containing food rapidly raises the blood sugar after eating with respect to a reference food, which is glucose or this white bread, depending. Common foods in Nigeria, we have the rice, we have the danwake, we have the tuwon masara, we have the tuwon semolita. All of them have a very high glycemic index, so indicating they raises the blood sugar from our results here. However, for the glycemic load also they have high content, even for between the danwaken flour and the danwaken dawa, which is less common in this part of Gombe State. It's only the pasta that is having a moderate amount of glycemic index. So most of the foods that we eat have high glycemic index and therefore raises the blood glucose level immediately after consumption. Another one that we have done is the analysis of vitamin A. When you come to buy your flour or sugar, you see that I logo on the bag, indicating it's been fortified with vitamin A. And this vitamin A fortification was made mandatory by NAFDAC and Standards Organization and of Nigeria. So we analyzed 4 samples of flour that is commonly consumed, and unfortunately the highest one is having only 8,000 international units per kilogram of the flour, having less amount of vitamin A. The recommended level is 20,000 international units and we have 8,000. So it may be as a result of many reasons. How we expose them when selling, because we just put them under the sunlight or anyway we just go and buy them. The temperature may even affect the vitamin A sample, or even the level of fortification right from the manufacturing. Another one is the vegetable oil. It's also mandated to fortify some of our vegetable oil or all the vegetable oil with vitamin A samples. Unfortunately, again, all the samples of the oil, that is the vegetable oil that we have analyzed, have low level of vitamin A. The highest one is having around 14,800 retinol concentration, and the normal concentration is 15,000. So as you can see, it also might be as a result of how we handle these products or lack of compliance. Next slide, please. We also analyze the antidiarrheal effect of what we call Jatrophacoccus, because mostly we still believe in this traditional medicine. This Jatrophacoccus is what we call beta da zubu in Hausa. So experimental animals were induced with castor oil to develop diarrhea, and they were given this particular Jatrophacoccus leaf extract. The highest is 400 milligram, which tends to be even more effective than the normal standard drug that is being used or recommended. We also analyze the effect of Carica papaya stem bark extract on the liver and kidney function test in experimental animals. So these experimental animals were made diabetic. After they are made diabetic, as you can see, the first one is the normal group. This is the diabetic control, and these ones are the groups that are being given this extra. Carica papaya, is Wanda. So the stem bark of Wanda was used. As you can see, the diabetic group has a high level of all these parameters. The aspartate transaminase, ALT, ALP, the urea biomarker, and the creatinine level is higher. So the effective giving of this stem bark extract, it tends to reduce. However, all of it have not reduced to the standard drug of metformin used in the management of diabetes mellitus. So in some researches, we've analyzed the hypoglycemic activity, the hepatoprotective activity, and the hypolipidemic activity of some of these traditional plants. We have the Vennonia amygdalina. This Vennonia amygdalina is called shuwaka. We have Guera senegalensis savara. We have this famous Adansonia digitata hookah, but this one is the seed palm. We have the Talferia ossidalentis, which is kabewa. And we have the Azadiracta indica, which is the famous name, and it acts to inhibit this schistosomial activity in snails isolated. So, Mr. Vice Chancellor, sir, the breadth of my research program has been made possible through deliberate cultivation of interdisciplinary collaboration with colleagues in other specializations. These partnerships has enriched the scope and quality of investigations, allowing for more comprehensive approaches to complex health challenges. So I am grateful to all my co-researchers and collaborators whose contributions have made this body of work possible. In conclusion, food is not merely a source of calorie. It is fundamentally a collection of molecules that interact with metabolic pathways. So every dietary choice represents a biochemical intervention. This lecture has covered the biochemical landscape of human health, from the molecular architecture of carbohydrates, proteins, lipids, vitamins, minerals, and reactive oxygen species, to their pathological consequences of their dysregulation, and the therapeutic potential of nature's molecular pharmacy. The study of small molecules, whether in food, blood, plants, or the environment, holds the key to addressing many of Nigeria's most pressing health and developmental challenges. The paradox of smallness, which I started this lecture with, finds its resolution here. Small molecules may be invisible, but their consequences in health, disease, national development, and in the lives of generations yet unborn are immeasurably large. Nigeria stands at a critical juncture. The country facing a dual burden of disease, which are the persistent high prevalence of infectious diseases, to the rapidly rising tide of non-communicable diseases such as diabetes mellitus, hypertension, cardiovascular disease, cancer, and malnutrition. Both of these categories are fundamentally biochemical in nature. So the future of biochemistry in Nigeria is therefore not merely a matter of academic interest, but that of national health security, food security, pharmaceutical independence, and economic development. One of the most significant constraints, however, facing biochemical research in Nigerian universities is the inadequacy of laboratory infrastructure. Without these tools, Nigerian researchers cannot conduct the experiments that will allow them to compete internationally or to solve the most complex problems facing their communities. So this requires not only capital expenditure on equipment, but also investment in maintenance culture, technical staff training, and the establishment of shared facility networks that allow multiple institutions to access expensive instrumentation. Mr. Vice Chancellor, my vision for biochemistry at this university is ambitious yet achievable. A department that produces graduates with internationally competitive biochemical knowledge and laboratory skills. A research program that generates evidence directly relevant to the health and nutritional needs of Gombe State and the wider northeastern region. And an outreach program that ensures biochemical knowledge flows from our laboratories into the communities and policies that shape the lives of ordinary Nigerians. So looking back across the over 15 years of my academic life, the journey has been shaped by curiosity, mentorship, institutional loyalty, collaborative generosity, and the resilience required to navigate the very real challenges of conducting research in a resource-limited environment. So every publication, every student supervised, every program introduced, and every policy engagement represents a step in a journey that is far from complete. Among all my contributions to academia, none has been more personally rewarded than the mentorship of students. Mentorship is not merely academic supervision, it is the deliberate cultivation of intellectual independence, scientific integrity, and professional identity in the next generation of scholars. I take particular satisfaction in watching my former students go on to secure appointments in various agencies and parastatals, some graduating to become medical doctors and pharmacists, some pursuing postgraduate degrees, and in some cases becoming lecturers themselves within and outside Nigeria. Foremost, I give thanks to Allah subhanahu wa ta'ala, in whose hands all knowledge ultimately rests, and whose guidance has been the light of this entire journey. This achievement would not have been possible without the love, sacrifice, and support of so many remarkable individuals, and it is with a full heart that I express my deepest gratitude to each and every one of them. I sincerely appreciate the Emir of Gombe and the Chancellor of this university for his gracious support and honor accorded to me. He has been my motivator and supporter since starting my PhD, and I pray Allah subhanahu wa ta'ala will reward you abundantly and bless you with excellent health, long life, wisdom, continued peace, and prosperity. To my parents, my nucleus, the bedrock upon which my DNA has been built. My father, though he couldn't come today. My father, the homeostasis of our family system. And my mothers, Adda and Gogo, your sacrifices laid the foundation for my education. And your prayers has been my most constant companion. You gave me the roots to grow and the wings to fly, and for that I am eternally grateful. To my enzymes and biological catalysts, Hajiya Dr. Aisha Umar Maigari, the former Commissioner of Education and the Commissioner nominee, the Wazir Hashidu Engineer Dr. Saeed, the Latu Hajiya Zainab of FTH, Dr. Yayam, many of them I have the lists here. I've seen Akliyu, Bidabo, all of you. You have been my greatest cheerleaders, Hajara, and my closest confidants. Thank you for always being my safe harbor throughout this demanding journey. I'll never forget the contribution of my late siblings, Hajja Usman and Engineer Abdul Qadir. May Allah forgive their shortcomings. To my buffer system and brother-in-laws, I can see Alhaji Sanusi Shehu Turaki, Alhaji Musa Yahya watching online, Alhaji Ahmed Bello, Alhaji Imran A. and Dr. Musa Abubakar, and Alhaji Salihu Ibrahim Hassan. Your support meant more than words can express because you stood by me just like my brothers. And your presence here today to share this milestone makes this occasion all more meaningful. To my stem cells, my beloved nieces, nephews, I can see most of you here. You may not necessarily fully grasp the complexities of this work that's led to this day. Nazi, you can do that. And your effortless ability to make me smile have carried me through the most exhausting chapters of this journey. Rafia, you have brought light, color, and journey into my world together with your other siblings, and this milestone is dedicated to the bright future of all my nieces and nephews, the bright futures that lie ahead of every one of you. And to my structural proteins, the family of Yarima Hashri, I've seen my uncles and that of Al-Alimo, the Bosarikim Malaman Duko also. This achievement belongs to me as much as it does to you. I am grateful to the Vice Chancellor, Professor Sani Ahmed Yauta, with whom I have the opportunity and privilege to work since the beginning of my career. The former Vice Chancellors, Professor Ali Usman El Nafati and Professor Ibrahim Umar, they shaped and guided me, particularly in my administrative role. Actually, what they did to me is I'm so grateful to what they have done because their influence will forever remain with me. I also acknowledge the present and immediate past registrars, Dr. Aliyu Bafeto, Mala Abdullahi Yuguda, and Mala Aliyu Kamara, whose genuine assistance has been invaluable. To Professor Hajja Gana Hamza, the Dean School of Postgraduate Studies, your guidance has left a lasting imprint in my career. I'm also grateful to Dr. Elisha Khan, the former Dean of the Faculty of Science, Malam Umar Manga, Malam Haruna Hassan, because they supported me when we were short of manpower when it was called this Wild Lady Department. I'm also grateful to Malam Al-Hassan Siddan and his family for their invaluable support. To my fellow metabolites and colleagues in the department, my two HODs, and the secretary, departmental secretary, Malam Sani, and all the staff in the department, their names are displayed, too many to mention. You are the secret behind every deadline met and success achieved. I appreciate also all the staff in the Directorate of Research and Development for their efforts in securing the research grants for the university. Nobody to mention is Malam Harisu. I also appreciate the staff, Malam Awal Jibrin from Subeb and Malam Adamu Aliyu and Hajia Zainab, because they showed me a life outside academia. So thank you for giving me such experience. I'm also grateful to the staff in the Vice Chancellor's office, the registry, the bursary, fiscal planning unit, and those in the university security unit. In particular, also I acknowledge Malam Ibrahim Abubakar Ifa, the deputy registrar, the academic secretary Malam Sarki Musa and his team. You have been present and helpful at every stage of my academic career. I also commend the University Ceremonies Committee under the chairmanship of Professor Muhammad Bura Maduku. Professor Oduku has been there, not aside even the Ceremonies Committee. He has been there and has been my motivator and supporter. And also Dr. Bappa, the former Dean Student Affairs. To all my research collaborators within GSU across Nigeria, the work we have accomplished together stands as a testament to what becomes possible when scientists share knowledge and efforts generously. So I am proud to have walked this path of inquiry alongside you. To the metabolic engines, my students, the past, the present, and the future, you are the reason this work matters. Every question you posed in class, every result you brought to me in the laboratory, every dissertation and thesis you defended, every career built upon this foundation have made my effort worthwhile. And also you are the living continuation of this work, and I could not be more prouder. My dear husband, Your Excellency, Sir, Ambassador Adamu Ibrahim, I am deeply honored to share this moment with you and for being the antioxidant in my life. To Aisha Nawal, my growth factor, thank you for being the extraordinary person that you are. I've seen lots of my cousins at the Altine, Musa, and see Antimbapa, Abubakar, I see lots of you, Auntie Sari, to thank you for coming and thank you for supporting me. Finally, I wish to thank the entire university community and beyond. Many individuals, too numerous to mention, for your contribution towards this achievement. Today, Mr. Vice Chancellor, sir, I represent every girl child in Gombe who wish to someday stand on this podium. I want you to know that with absolute certainty, as you watch me stand on this stage today, there are no limits to what you can achieve. To every father who wishes to have his daughter take this path, I will encourage you, advise you to stand by her and motivate her, because my father has been my backbone and #1 fan. So, I can finally say, and proudly say, I have been standing on the shoulders of giants. Alhamdulillah, fasubhanaka Allahumma wa bihamdik, ashhadu an la ilaha illa anta, astaghfiruka wa atubu ilayk.
Nigeria today, great! Nigeria today, allow me to say, allow me to yarn, allow me to yarn, allow me to yarn, allow me to
Thank you very much. Please take your seats. Please thank you very much. Thank you very much. This is really the learning of our personal
Alhamdulillah. Alhamdulillah. We have heard it from Professor Fatima Maigari. And now we need to go to the next item on the program. And may I respectfully invite the Vice Chancellor to please come up here for presentation of award to Professor Fatima Maigari. And let me also invite Professor Fatima to please come up here.
Distinguished gentlemen and ladies, this is an award of excellence from Gombe State University for Professor Fatima Umar Maigari, BSc, MSc, PhD, MPA, for presenting an inaugural lecture on the 30th of June 2026. Congratulations.
Thank you.
Gombe State University, this is to certify that Professor Fatima Umar Maigari of the Department of Chemistry has presented the 25th inaugural lecture titled Small Molecules as the Biochemical Architecture of Human Health on 30th June 2026. Congratulations, Professor.
Thank you.
While he takes our seats, we quickly revise the brief of the school. We will come to the word of God. We have a applause for Professor Robert. We will now be of the science who is coming for the class.
The Chancellor of this great university, the Vice Chancellor, ably represented by the Deputy Vice Chancellor, Admin, the Deputy Vice Chancellor, Academic, all the principal officers here present, members of the Senate of this great university, the chairman and the members of the ceremonial committees, special invited guests, relevant relatives of Professor Fatima Umar Maigari, our royal fathers here present, good evening. It is my pleasure and honor to stand before you here to give vote of thanks on behalf of staff and students of Faculty of Science, where Professor Fatima Maigari is a member. When I look at her title of her presentation today, Small Molecules, I start thinking, is it proof because you are small in size? That is why you come out with a small? What is not small in size, but big in name? She has contributed a lot, and she is one of the architecture of the Department of Biochemistry. I congratulate you especially for this important occasion, for your achievement in the academia. Your name is written in the academia environment. To the Vice Chancellor, sir, I thank you very much for providing all the inaugural environment to host this event. To my fellow colleagues in the Senate, deans, directors, heads of departments, I say thank you very much for being here to support Professor Fatima Maigari. To our invited guests, I say thank you very much for sparing your time to grace this occasion. You have a lot of engagement, but simply because of contribution of Professor Fatima Maigari to the academia and to the national or to humanity, you have reserved the time to be part of this occasion. I say thank you very much. To the immediate family and the extended family, this occasion does not belong to you only, it belongs to Faculty of Science. Together we have made it a reality. I say thank you very much for your coming. To the ceremonial committee, my mentor, Professor Muhammad Gurama and his committee, I say thank you very much. As we live here to our various destination, it is my sincere prayer and hope that every individual will reach home successfully. Thank you all and God bless.
Thank you very much. Thank you. The round of applause once is for the First speaking officer, immediately after national